Yes.
Okay, great. Okay, thank you very much to the organizers for inviting me to this wonderful event. I'm Chairman Kim from the nearby city of Ulsan. For the finale of the last session, I'll be talking about the biosynthetic cost for the proteins to make you sleepy at night. Okay. So this is the widely accepted pattern of oscillation we all get into in a daily basis. The sleep hormone, melatonin, denoted as a green line here, starts to rise sharply around 9 p.m. and quickly takes over your body unless you binge drink caffeine or Red Bull. The local time here is at 10:20 p.m. This time yesterday, fortunately, we had wonderful and exciting talks by Shlomi, Tegun, and Joachim. Today, I'm not sure if I could keep you awake or leave you under the influence of melatonin spree, but let me try my best. Okay, my talk today consists of 3 parts: a bit of introduction to circadian rhythms and the underlying cell biology. Then follows a sort of compilation of my recent works and others on the cost of biological rhythms. For that, I'll try to rationalize the post-translational modifications from the perspective of cell economy. And I'll wrap up my talk with a short summary. All right, this is a single slide for a quick review on the circadian oscillation. Okay, so the word circadian comes from the Latin circa and dies, meaning approximately a day, 24 hours. Number 2, the rhythm is maintained by daily ups and downs in cellular activity or concentration of gene products and metabolites, even in the absence of external cues. Number 3, it is ubiquitous across the organisms. Number 4, the rhythm is influenced by external signals, including light and food intake. Interestingly, though, there are temperature compensation mechanisms so that the periodicity is not much swayed by the change in temperature. Number 5, it impacts every possible physiology of animals and plants. And last but not least, its disruption not only causes a sleep disorder, but also may lead to serious diseases like cancer. It is not a pet peeve for shift workers. It's a serious health issue. All right, then the natural question follows. How is this daily cycle generated? The dynamical system under external restoring force causes the oscillation around an equilibrium. Then the maintenance of oscillation is a matter of balance between the external driving and the dissipation. In Peter Holmes' talk this evening on calcium oscillation was driven by autopoietic influence hierarchy and some other factors. However, in other circumstances, the oscillations are maintained by interactions between components. Think about the Lotka-Volterra model for predator-prey systems. For circadian systems, specifically for mammalian, the core machinery is the genetic regulation like this. The so-called clock genes are produced under the influence of the transcription factors whose activity is controlled by the clock protein themselves, okay? And this is a plant version of the circadian system, specifically for Arabidopsis thaliana. Not surprisingly, the plant circadian system is much, much more elaborate and sophisticated, containing a lot more players than its mammalian counterparts. As you see, in the heavy negative feedback loops, whose prevalence might be linked to evolutionary rationale. But here, I'd like to emphasize that the circadian system is a negative feedback system from the control perspective. All right, Again, the circadian system is by construction negative feedback with intermediate time delay components. And the production of circadian proteins follows a temporal modulation. All right, so the simplest model to describe the quantitative aspects of circadian system starts with the rate equation like this. Proteins are expressed constitutively or in a regulated manner, but they are subject to degradation. Here we denote time-dependent protein concentration X of T, the synthesis rate GT. It's a certain periodic function because of the previous mechanisms I mentioned. And it is determined by the details of involved genetic regulation circuitry. And the degradation constant R here is allowed to have time dependence. So RT in general. So once we employ the previous equation of motion, simple birth-death-based equation of motion, we can say quite a significant constraint on the degradation rate simply by requiring each parameter or variable to be physically sensible. Because it's nonsense when these quantities become negative, we require R of T cannot be negative, right? Since RT is also non-negative, RT cannot be lower than this quantity, right? So RT cannot be lower than the maximum of the negative of the so-called sort of a fractional changes in XT over time. So this inequality should be satisfied for all time T. Now, if you are going to infer a constant degradation rate R instead of RT from empirical data, which is a common practice in modeling works, the rate should be lower bounded by the maximum of the fractional change in XT. So we can define our minimum as the maximal value of this. Here we have two distinct waveforms. These two may well lead to a different value of our minimum, which can be interpreted as different half-lives. On the left, you see a slightly lower degradation rate, meaning a longer half-life, right? So wide shape here, wide shape give relatively long half-life, while the narrow ones shorter half-life. So depending on the overall qualitative feature of the waveform, we can have a sense of how long the half-life of the protein will be. Okay, to complete the daily oscillation, the balance between the synthesis and degradation matters. For a given synthesis program, a periodic oscillation in XT becomes possible only when the degradation rate perfectly balances the synthesis as shown in the rightmost column, okay? Again, once the oscillator signal is given, and if you model the underlying dynamics as a birth-death process, we can obtain the lower bound of the constant degradation rate by looking at the waveform. As a rule of thumb, the sharper, the narrower signal indicates the higher degradation rate, meaning shorter half-life of the protein. And this is actually, this speculation is actually supported by the empirical data. For plant circadian protein PRR5 and PRR7, the previous statement consistently explains the relatively shorter half-life of these two proteins. These rate values are way higher, actually. Or the corresponding half-lives are unusually short. Some proteins, if not all, indeed show a sharp, non-harmonic, almost like a triangular waveform. And this requires a high level of degradation rate or a very short half-life, which is a big problem from the cell economy point of view. All right, it seems a really endless, futile task if we have to produce something making use of expensive raw materials and energy for use in a short spell and have to break them down again and again. If we add up all this product produced and broken down, that will be really a situation where many little makes a mickle, tickle more tesan. So the only solace from this analogy is, the only solace from this analogy is, unlike the Greek mythology, there is an end, which is, of course, death of an organism, okay? We are all mortal, so this game will come to an end eventually. So by definition, a periodic rise and fall comes back to the original point after a cycle, right? Thus, no net change in concentration xt after a period t, okay? So because of all the newly synthesized proteins, or exactly the same amount of protein to be precise, had to be broken down, this should be satisfied. At this point, we can define the cost function chi from the kinetic profile of an oscillating signal, which is either these two integrals, right? So once the time series xt is given empirically or reconstructed, then we can determine our minimum and by using this value, we can compute the cost of this biological rhythm, okay? According to the ballpark estimation, daily production and breakdown of PRL7 is 6 to 7 times more of its average amount of in a cell, okay? Then comes a question. This picture may be an artifact because we are assuming a constant degradation rate. What if the degradation rate is time varying, okay? That may be an exit to escape from, or at least mitigate this doomed Sisyphean task. All right, indeed, if we introduce the temporal modulation of the degradation rate, the overall cost would be reduced, as you see here. To generate this periodic signal xt, we have continuous spectrum of choices for GT and RT, okay? So if we compare the brighter pink and sky blue combination to the darker red blue pair, the darker pair saves the production cost to a significant degree. Then the question follows, how? How does a cell realize the time varying degradation rate? Okay. So employing another agent, another designated protein responsible for rhythmic degradation of circadian protein, that's probably an overkill. At least it doesn't address the problem of cell economy, right? So a natural way of introducing the oscillating RT is by way of ubiquitination proteasome pathways. Actually, the role of ubiquitination in many circadian proteins are indeed well established through numerous experimental and theoretical studies. Probably we need another talk for the details of that. So to make a long story short, the ubiquitination pathways are conducted by 3 major actors: ubiquitin, E3 ligase, and deubiquitinating enzyme, or deubiquitinase, all right? So roughly speaking, ubiquitin is a small peptide, 76 amino acids, something like that, consisting ubiquitously found in eukaryotic cell. That's why the name ubiquitin. And the ubiquitin is sort of a death tag for a substrate protein, okay? And E3 ligase is a kind of helper or chaperone. Chaperone is reserved for another sense, but yeah, it's a helper in attaching the ubiquitin molecule to the target substrate protein. Deubiquitinating enzyme may bind to the ubiquitin-bound substrate to dissociate the ubiquitin from the substrate protein. So the possible scenario includes either the degradation along this pathway or salvation and recycling of ubiquitin along that path. Right, the molecular details are slightly different species by species, but the core network or reaction pathways are pretty well conserved and can be modeled like this. The bare substrate protein, clock protein, X0 is reversibly bound and unbind with E3 ligase, like you see here. The substrate plus minus 0 stands for the protein's binding status with respect to ligase, deubiquitinase, and neither. So X0 meaning neither of the enzymes are bound to the substrate protein. The presence or absence of the star asterisk superscript indicates the binding status with respect to the ubiquitin. Okay, so remember, ubiquitin binding is irreversible. Okay, whereas the binding and unbinding with an enzyme, E3 ligase and deubiquitinase, is considered reversible in the scheme, which is usually followed by modeling works. All right. So to describe the ubiquitination pathways in simplest possible method, we set the system of ordinary differential equations. So by adding the terms side by side from this, okay, this starting set of ODEs can be transformed. Okay, so by adding up all the terms side by side, first we get the equations of motion for the total concentration. Xt is the sum of all different forms or different compartmentalized form of substrate protein. So we remember, if even though we are classifying, categorizing the substrate protein into different forms, we still have a constant rate R0 for the turnover of the protease activity. Then the transform of the system of the ODEs lead us to the explicit form of GT and RT, which is again fed back to the computation for the biosynthetic cost. Eventually, the computation of chi. Okay, so contrary to the conventional ODE-based simulations, where we start from the rate function and predict or explain what we will detect or what has been already observed, right? But this method, I'll largely skip the details. So-called, we named it as backward simulation, which was published in separate paper a week ago. So yeah, contrary to the conventional simulation method, we start from the empirical data x of t, okay, the empirical kinetic profile to arrive at the rate functions that fits the effective birth-death dynamics. So the technical details can be found in this paper, which was recently published. The point here is we needed some decent and reasonable techniques to deal with many variables and unknown kinetic parameters. And specifically, we had to cope with minuscule of experimental data available. which is a common problem for the modelers. The essential part is the iterative fitting of the experimental data obtained in coarse-grained manners. For example, the empirical data available for the concentration of substrate protein is the total concentration instead of the compartmentalized forms like X naught, X plus, X minus, and so on. So to reconcile the marginally or minimally available experimental data with no inconsistency, that's the bottom line of this methodology. So how does a temporal modulation of RT arise from ubiquitination? So we tried an intuitive explanation in the paper published last July, and this is an excerpt from the paper. So when the synthesis is very active, more and more newly produced proteins are floating around in cytosol without having a chance for the kiss of death, right? So a relatively small fraction of protein substrate is along the degradation pathway. On the other hand, when the synthesis is less active, minimally active, more protein molecules are already ubiquitinated like this, right? So even with a constant reaction rate R naught, the overall or effective degradation rate R of T tends to be working in approximately opposite phases to the synthesis activity, okay? Shortly, we will see the detailed kinetic profiles demonstrating this complementary nature between synthesis and degradation. So another point to make is related to another form of post-translational modification, okay? So it is well known that the ubiquitination pathways may well depend on the phosphorylation as a kind of preparation for the following processes. To reflect this aspect, we introduced an extra reaction step in our dynamical model. So instead of direct ligase binding with a bare substrate, we introduced the phosphorylation, dephosphorylation reaction. Here, capital K and capital F, respectively, is the intracellular level of kinase and the phosphatase, dephosphorylating enzyme, respectively. And accordingly, the recycling of the substrate here is rerouted to the phosphorylated form instead to the bare form. Then the resulting set of equations of motion is given like this. The green parts are, of course, the revised kinetic terms. On top of that, to simulate the multiple phosphorylation, multi-site phosphorylation, you can add up to arbitrary n extra steps according to the number of phosphorylation sites on the substrate protein. So you can increase the dimension of this dynamical system up to 7, 8, as many as you like, okay? So the oscillating protein abundance could be realized by various combination of synthesis and degradation function. You can clearly see the enhanced rhythmicity as we increase the number of phosphorylation sites. So here N means the number of phosphorylation sites on the surface of substrate protein. N being equal to 0 means it's ubiquitination only, okay? So phosphorylation comes first and ubiquitination follows. That's the scheme, okay? So as I said, N is the number of phosphorylation sites existing on the surface of the substrate. Even though the ubiquitination itself was capable of creating temporal modulation of the degradation rate in phosphorylation independent manner, this added extra steps of phosphorylation further enhances the rhythmicity of the degradation process, okay? So, and saves protein production cost chi, right? So ubiquitination only still generate temporally modulating degradation rate, which is good. But if you add more extra steps of post-translational modification, then yeah, you could save a lot more. Okay, to summarize, if you're still awake, number one, we found that the post-translational modification of circadian proteins naturally gives rise to the temporal modulation of degradation rate, thus RT. Okay. Number two, added steps of extra post-translational modification, which is phosphorylation, in our example, further contributes to the cost-efficient maintenance of the circadian rhythm. So all these works largely contained in these two publications. And finally, as a final remark, all the works presented in this talk were done by Jung-Eun Che, my graduate student, and Dr. Lim from Hong Kong Baptist University. And Professor Pan-Jun Kim has long been a tireless, fearless, and motivated collaborating partner of mine. And we are grateful for the financial support from NRF, HKBU. We also acknowledge the generous resource support from our affiliated supercomputing centers. Thank you very much. Did I finish too early?
Yeah, 10 minutes. 10 minutes.
Yeah.
I
have to say thank you very much to the audience for awaiting my turn.
Nobody is asleep.
Okay, that's
Everybody
good.
is wide awake. Okay.
Thank you very much for the nice talk. I'm not sure that I clearly understand how we can make the oscillation of the protein level, but it seems that you add some protocol or some reaction rules to make the protein level oscillates in time. So I wonder that for other proteins, probably we need to keep a certain level compared to other circadian rhythm. So I'm wondering that then if there are two circuits or the chemical reaction mass to produce oscillating protein level and the other produce the constant protein level, then do they have topologically different reaction mass? Can we detect? Can you hear me? Yeah.
Okay. So, okay. Thank you for the nice question. And to answer your first question, I don't have any special recipe except that we were using this law of mass action. So as you have seen, all the mathematical method was based on ODEs. Even it's not stochastic approach. We didn't mention anything about master equations, Brownian motion, right? So, but in a sense, this temporally modulated reaction rate is a natural outcome of reaction networks. As we have seen in, okay, I'll show you. Yeah, as we have seen in this network, what we are measuring in the laboratory is actually the sum of all these components, right? So even though individual respective form of ubiquitinated protein substrate, they may be under constant degradation, but overall, because of this transforming of the conformation, overall, the sum of all this substrate protein gives rise to time-varying degradation rate. But that is my first answer to your first question. And what was the second one?
My question is about, can we predict that they give the oscillating degradation rate only by looking at the structure itself?
Oh, structure. You mean the
like
molecular conformation or network structure?
Yeah, this structure, current slide. So if you look at this structure of the X0 and how they can transition to the.
Oh, yeah.
Only by looking at,
Yes. Yeah, in principle, yes. Yeah, the simplest example is probably the mutually, so as you see here, this is made up of basically a two component with the time delay. So one is activating, one component, did I miss something? So one component is actually activating the other, while the other component actually represses or suppresses the activity of the previous one. So in that way, you can keep this overall negative sign of the loop. Okay, negative sign meaning it's in a sense it's inconsistent. Okay, if you constitute your system with 3 mutually repressing proteins, then that's naturally generating oscillating signal. That's a celebrated example of repressilator, right? So it's like a rock, paper, scissors situation. So in that sense, yes, if we have decent information on topology or reaction network structure, yes, we can say at least it is possible, plausible for a certain protein to show temporally oscillating kinetic profile.
Thank you very much.
Thank you.
Thank you for the nice talk. I'm Jung Chek from Busan National University. So can you hear me clearly? Or
yes, thank you.
So I have basically two questions. So the first question is related to the modeling part. So as you mentioned in your presentation, typically these kinetic models require a lot of parameters that cannot be determined experimentally, right? And you mentioned that you use some sort of constraints to guess the values of those parameters. But yes, I think still you will get a huge degeneracy of parameters. So you have to use some sort of, well, educated guess for those parameters, right? So I just want to know more details about how you determine those values. That's my first question. And the second question is kind of related. I think currently proteomics can detect those post-translationally modified proteins separately from the unmodified proteins. So if you have this information, then I think you can improve your model, right? Because here you just know the whole concentration of some species, but you cannot separate those contributions from different PTM states, basically, right? So I wonder if you have any plan on this kind of direction. That's my second question.
Okay, thank you for the constructive comments and question. Yeah, to answer your question, yes, it's really a curse of a huge ambiguity innate in kinetic parameters. Yeah, as you correctly pointed out, we cannot, there's no royal road or any magic recipe to get over this difficulty. So our magic was basically the experimental data. We iteratively use the time series XT. Okay, so basically we have a couple of discrete data points from which we reconstruct a spline. And then, to make a continuous time series data to work with differentiation and so on. And based on this empirical data, to make a long story short, all the details are actually stipulated in this paper. And we run several rounds of feedbacks. So we check the resulting signal is correct or not, and goes back to the previous sanity check step to check whether any crazy parameter led us to a crazy situation. And this is pretty messy and non-systematic way, I have to confess. But once you find the set of at least relevant physiological parameters, at least you have what you can say. So that's what I can say. And the second question you asked about the kind of compartmentalized forms of different, the same protein. We are all theoreticians here, and I'm probably the last person to talk about spectroscopy or crystallography on other experimental technique. But so unless there is, to be honest, it's a matter of technological advance in experiments. So one story I recently heard is, yes, one big difficulty barrier in determining the crystal structure of protein was basically the membrane proteins, insoluble proteins, are really hard to crystallize. That's the good excuse on why we don't know much about the protein structure. But these days, I forgot the detailed name of the technology, but somehow people could make the crystals of insoluble membrane proteins to churn out the crystallographic data from Pohang accelerator and so on. So anyway, I have no deep insight on that, I have to confess. But yes, what I can do is by looking at the detailed biochemical pathways from literature and from model works, at least we can say about the plausible mechanisms behind the cell economy like this, okay? So if you imagine the wasted resources by simply constantly degrading the protein, that's a huge amount, right? So simply by introducing the time modulation, temporal modulation of the rate parameter, we can easily get over that difficulty. I mean, the cells or living organisms can do that. So finding the rationale behind this temporal modulation was, it was fun, actually. It took a bit of time. It's in a sense, it's a sequel work to the previous publication by Hang-Hyun Cho and Panjun Kim. Actually, we learned a lot during the collaboration process.
Yeah, I think this work is valuable and it gives a lot of intuition and insight for the community. So, but I just had some comments and suggestions. So thank you for your answers.
Thank you very much.
Okay, no more questions? Do you have a message?
Oh, yes, I will check. So where is, oh, I don't see any chat message.
Yeah, no questions in the chat.
Okay.
Okay, so thank you everyone for contributing to this session. Now we have